So we're going to talk about the structure of DNA today and what's going to happen in our lessons. So you're going to need to know how DNA is structured. You're going to need to know Chargaff's rules. You're going to need to know how base pairing works. And all that's going to be done today in our first lesson. All right, so the structure of DNA and what it is. All right, so DNA is really unique, guys. You have to think about this. So DNA is the one substance that all life is based off of. There's a large amount of differences between your genetic code and the genetic code of a frog or a tree or a snail. There's so much variation, but yet it's only one molecule. And so the question is, how can that be possible? How can that be true? And the answer lies in the structure, okay? So you should be aware of that. So the structure is important for understanding how the genetics are transferred, okay? So we're going to talk about how those genetics are transferred from one to the other based off of this one genetic molecule. All right, so the components of DNA, you need to know what they are. All right, so DNA is a nucleic acid. Okay, that's what the name of the molecule is. They're joined by long strands of covalent bonds. So long strands of covalent bonds stacked one on top of the other is what makes a nucleic acid. So each strand is a nucleic acid. So with the DNA structure, there's some words that you need to be aware of. You need to know what nitrogenous bases are, sugar phosphate backbone, and base pairing. All right, so what this means, so you have these two twists, right? It looks like a ladder that someone has just twisted, okay? That's the double helix. So helix just means twist. So double helix means there's two twists. All right, so you have this sugar phosphate backbone. That's like the sides of the ladder. And the base pairs make up the rungs. All right, so what these base pairs are adenine, guanine, cytosine, and thymine. All right, those are the nitrogenous bases. Nitrogenous. You should look at that word and realize that it says nitrogen in it, right? So they all contain nitrogen. All right, so nucleic acids are these long strands of DNA. All right, they're originally identified in these cell nuclei. Those are what we've been picking out. Those are what were visible in the chromosomes when they're condensed. So these nucleic acids are what make up DNA. That's just the name of the molecule for DNA. That's the type of molecule it is. So the nucleic acids are made up of these nucleotides. And it's like a bunch of them on a strand. So it's almost like pearls on a necklace. Lots and lots and lots of single pearls, lots and lots of nucleotides make up a nucleic acid. It's like a strand of these. All right, so it's just a string bound together of nucleotides. Lots of them bound together is a nucleic acid. One by itself is a nucleotide. So it's adenine, guanine, cytosine, and thymine. Those are the four that you should know. So these nucleic acids, these nucleotides anyway, are made up of three components. You have a phosphate group, which makes up the side of the ladder. So you have the phosphate attached to a sugar ring attached to the base. So you have a sugar phosphate backbone with a base stuck on the end. Now the base varies. They're named after their bases, okay, the nitrogenous bases, and they're rings. So what you're going to see is cytosine is a single ring. Thymine is a single ring. Adenine and guanine are doubles. All right, so there's two rings that are stuck together for both of those. And they wind up being in two different groups. Don't worry about uracil over here, this one over here just a second. We'll get to that in a minute. But the four main ones are going into two groups, purines and pyrimidines. All right, so they're joined by covalent bonds. So these pearls, these nucleotides on this string are joined by covalent bonds. So a covalent bond is one of the strongest bonds. A covalent bond is a true bonding. So if you haven't had chemistry yet, you're not really sure what that means. So it means that they are shared. They share electrons. It's like holding hands that can't be broken, okay? Those hands are really, really held together. Can I break them? Yes, but it's going to take a lot to do so, right? Covalent bonds are really strong. So a covalent bond is linking all of these right there at that sugar phosphate group. So this bond is the one that we're talking about, which is the one that's right in between each nucleotide. That one, that one, that one. It's what's really holding your DNA together. Oops. Gotta go back. All right, so we have these two groups. Pyrimidines are the single, and purines are the double. The way I remember it is the singles have the longest name. But there's these four types: cytosine and thymine, and adenine and guanine. And they always work where one of the purines attaches to one of the pyrimidines. So you always wind up with a double touching a single, a double and a single, a double and a single. And the way they work is A to T, G to C. All right, so they stick out sideways from that chain, so they wind up being in the middle, like the rungs of this ladder. All right, so you need to be really aware of this big idea. This is the one that they're going to test you on. You need to know that any sequence of bases is possible. Because there's four separate nitrogenous bases, you can arrange those almost in an infinite amount of ways. As long as the DNA gets longer, they're completely unique. That's the reason why DNA is that single molecule that all life can be based off of. Because all that variation can be accounted for in the length of DNA and the changing out of these nitrogenous base pairs. That is why DNA is used by almost every organism on the planet. All right, so you also are responsible for knowing how we solved this mystery. When we started out in the scientific world, we knew that Gregor Mendel was aware that something had to pass down from the parents, and he knew that it had to be half from mom, half from dad because of the way the genetics worked out, right? But what he didn't understand was the DNA part of it. And we didn't understand that for a very, very long time, all the way up to the 1800s and then into the 19th century, where we're trying to still work out what in the world is getting passed on. So in the 1900s, we found a couple of different scientific experiments that led to our understanding of what DNA actually looks like. We found that what it was, and then we had to prove that it was that, and then we had to figure out what it looked like. All right, so there's Franklin, Watson, Crick, and Chargaff that are the big scientists in this. All right, so the first one is Erwin Chargaff. And what he found is after we discovered that DNA was most probable the genetic carrier, we didn't know for sure, but we were most probable for it. And so what happened is Chargaff said, well, we're going to do these experiments. And what he found out is he can test the presence of these nitrogenous bases. And he said, okay, every time I look at this molecule, what's really weird is that the exact same amount of adenine is always the same amount of thymine. And then if he looks to cytosine, it's always the exact same amount as guanine. He had no idea why. But he knew these four nitrogenous molecules, he didn't know they were bases at that point, but these four nitrogenous molecules, what he found out is they always matched. So he came out with Chargaff's rule, and Chargaff's rule says for every A, there's a T, for every G, there's a C, always, and nobody knew why. All right, but that's Chargaff's rules. He was just the first to accurately measure nucleotide amounts, and it kind of led to a bunch of questions, and then a bunch of scientists decided to start studying those. because the structure of DNA was one of the greatest discoveries of our time. Along comes this unsung hero. Her name's Rosalind Franklin. So Rosalind Franklin was known to be a little, I don't know how to say this in a nice way. She was a little pushy, but as a woman in science before the 1950s, we're talking like really, really a boys' club here. Women weren't even allowed to get master's degree or hold any degree of any kind until 1947. She worked in 1940 to 1960. She was in that age and era where women had like no rights, and even when they did have rights, nobody really wanted to give them anything. Okay, so just be aware that's the chauvinistic era that she was in. All right, but in 1950 she was an X-ray crystallographer. So what she did was she took photographs basically of very, very tiny things using radiation photographs. So it's called X-ray diffraction. And basically, she accidentally took a photo of DNA. And this is that photo. This is photo 51. It's super famous. So she famously did not get along with her partner, who was pretty much a chauvinistic guy. He did not like working with a woman. He did not want to. He made that clearly known in the scientific community. They did not get along at all. They butt heads because she was a strong woman, and in an age where strong women were not accepted. And so what happened is her supervisor and her partner was Wilkins, and they did not get along. And so unfortunately, they didn't share their data with one another very easily. And then the university they were working with was in England. It's called the University of Cambridge University. And Cambridge University had multiple teams of scientists working on the same idea of DNA structure. And Rosalind Franklin was kind of looked over because she was the only woman. When she had found that photograph, it was 18 months prior to Watson and Crick. And Watson and Crick are the ones that have been given credit with discovery of the DNA structure. And the reason why is kind of a little messed up. All right, so Rosalind Franklin did not work together with her partner, but Watson and Crick were two men that actually really worked well together and worked well within the university. And so what happened is when she discovered this, it was a full 18 months ahead of the others, she gave a talk at the university, which Watson was present in the audience and suggested, based off of her photograph 51, that it had to be most probably a double helix with base pairing in between. And just off the cuff said that, and that was it. And then lo and behold, a few months later, Watson and Crick got a hold of all of her data. Her partner and lab supervisor gave her data that she had finished and gave it to Watson and Crick without ever asking her permission. Was it illegal? No, because they were her supervisors. Was it nice? Absolutely not, right? And so what happened is they wound up publishing a paper using her data to back up their ideas. They didn't do any experimentation that actually worked. All their experimentation was based off of was hers and did not give her any credit. And the worst part of it is she died at 37. I'm 37. She died at my age of ovarian cancer because she was handling so many forms of radiation. She was taking these radioactive photographs every single day, all the time, and they were not aware at that time of radiation issues. And she died of ovarian cancer at a very, very young age, quite literally died for her work. Watson and Crick, less than 4 months after she died, won the Nobel Prize. didn't even mention her name. She was not allowed to get or receive the Nobel Prize because it's never given posthumously. It's never given after death. So messed up. All right, but she will never win a Nobel, but everybody is finally giving her the recognition she requires. In fact, he famously said a bunch of nasty things about her in his memoirs. After his death, his memoirs were published. I guess he waited till after his death. And he talked about her in those memoirs and said she was a cantankerous woman. I don't think they liked her very much, but that doesn't give you the right to steal her information and without giving her permission, right? But these these three men here, Crick, Watson, and Wilkins, her partner, wound up with the Nobel Prize for discovering the structure of DNA. I messed up. But again, she did not give them any permission. It was all done on without her permission, which is kind of messed up. And this is what he wrote in his memoirs. Clearly, Rosie had to go or be put in her place. The former was obviously preferable because given her belligerent moods, it would be very difficult for Maurice, talking about Wilkins, to maintain a dominant position in the lab. I know, kind of messed up, right? But it tells you the situation that they grew up in and the situation she was working in. Obviously, she was a strong woman who had great big opinions in a world that women shouldn't have an opinion, is what they thought. All right, but Watson and Crick wound up getting credit for that model. And that breakthrough model was important because it showed why Chargaff's rules worked. It was beautiful. It worked with it. All right, two strands are wrapped around one another in that beautiful structure. And what does it tell us? It tells us that it explains why Chargaff's rules of base pairing worked and how they're actually held together, because DNA must be together and apart all the time. It has to do both, which was really confusing to a lot of scientists because we can imagine it being this long strand of things, but two strands that separate was a completely different idea at the time. All right, so again, it looks like this twisted ladder. It's a double helix. It's almost like a spiral staircase, you know, those pretty staircases that you walk up. And the two strands of DNA are held together by these base pairs in the middle, A to T, G to C. And the one thing that they figured out is, in fact, came straight off of her paper. It was that she said that it had to be an anti-parallel strands. So you know what parallel means, right? Together, like this. Anti-parallel means like this. And that's what she was saying, that it's what we call 5' to 3', the 5 carbon to 3 carbon on one side. And the other side is the opposite, 3 to 5. It's backwards. So when they're running transcription translation, they're running in two different directions like a highway. One's running down, one's running up. All right, so it allows each strand to carry a sequence of nucleotides. They're like letters of the alphabet. That alphabet describes proteins. Proteins make up a person or an animal or whatever it is, okay? So DNAs, nucleotides code for genes, which code for proteins, which make the being of whatever it is, the organism. And they're anti-parallel, backwards. The other thing it explained is how those two strands were held together, but yet can come apart pretty easily. If it was a covalent bond, it would be very hard to break apart. Covalent bonds don't break normally. All right, not easily, not without some sort of chemical reaction, which would destroy DNA. And so what they found is these hydrogen bonds had to hold it together. So we talked about hydrogen bonding when we talked about how water worked. Remember we said water is polar and the H and the O kind of get sticky together. And that's what's happening here is it's polarity. So what's happening is it's kind of like little magnets. You can get them close to one another and those magnets are just going to attach, aren't they? And that's what's happening is these attach with these hydrogen bonds and they're a difference in polarity. Positive touches negative, they're going to touch like a magnet. And that's what happens. But you can pull magnets apart relatively easy, and that's what it is. It's basically held together with these polar areas here, which are just hydrogen bonding. So it's almost like held together with magnets. You can separate it, do what you need, and it comes right back together again. And that's kind of beautiful. That's how it's working. All right, so the base pairing, like we talked about, A to T, G to C. That's why Chargaff's rules were coming around, is because for every A, there's a matching T on the other strand. And for every G, there's a matching C, right? A to T, G to C, always. Memorize that. It's an important step of information that you're going to need to know from here on until you finish taking any science class forever. A to T, G to C, adenine to thymine, guanine to cytosine, and that is why Chargaff's rule always showed up. We didn't know why at the time, but we do now. We know that that's where they're attaching. Every A to every T, every G to every C, that's what's attaching. And what you wind up is if you look over here with the adenine and the guanine, you can see the base pairing. So A to T has 2 hydrogen bonds holding it together. G to C has 3 hydrogen bonds holding it together. It's like 3 magnets versus 2 magnets. So it's perfect, and it explains Chargaff's rules. So this is why Chargaff's was coming out. We didn't understand it. A to T, 2 hydrogens. G to C, 3 hydrogen bonds. Okay? That's just the way they're structured, which is pretty awesome.